Edwin Allen and his research group constructed a study into Keratin 12 missense mutations and the effect that they have on the cornea. This produced the paper, Keratin 12 missense mutations induces the unfolded protein response and apoptosis in Meesmann Epithelial Corneal Dystrophy. Missense mutations occur from a substitution of an amino acid caused by the change of one base pair. These can potentially increase the effectiveness of a protein. However, the missense mutation in Keratin 12 results in the protein unable to polymerize. This results in Meesmann Epithelial Corneal Dystrophy. This is a disease associated with the eye, which forms small cysts on the top layer of the cornea. These cysts can occur at any time in life but don't cause many symptoms until they burst and cause irritation. This will ultimately result in an increased tear production, sensitivity to light, also known as photophobia, the feeling of having a foreign object in the eye, and sometimes having episodes of blurred vision. In patients with corneal dystrophy, there is evidence of 14 mutations in the Keratin 12 gene, 10 of which are substitution mutations identified in helix initiation and 4 in helix termination. Such motifs are important for intermediate filament formation and assembly, as they determine intermolecular interactions which will influence protein folding.
In this disorder, there's a mutated Keratin 12, and Keratin 12 is expressed in the eye, in the cornea of the eye, the outer layer of the eye. And keratins normally form long polymers known as intermediate filaments. In this case, Keratin 12 is mutated, and so it can't form the polymer.
In the experiment, mice were bred to express the protein Keratin 12. In order to do this, the mice had to contain the human mutant gene. A small section of the human DNA containing the desired gene was taken and edited before being inserted into the mouse targeting vector. Homozygosity was bred into the mice so that the altered genotype would be expressed in the mouse's phenotype. PCR was used to determine the genotype of the mice using DNA from ear biopsies. The mice were then euthanized through carbon dioxide asphyxiation and their eyes were snapped frozen. The eyes were stained with fluorescein, then imaged through slit lamp assessment. The corneal surface of each mouse was evaluated at 7 weeks. Whole eyes were enucleated and fixed before the corneas were dehydrated and imaged. The experiment involved the creation of a mutant mouse allele L132P. However, the expression of this allele in the mouse did not show the phenotype.
In heterozygous mice, you have one copy of the wild-type mouse allele and one copy of the mutated human allele, and that should have represented the disease phenotype. So people who have Meesmann's, they have one wild-type copy and one mutated copy, and then they have the disease phenotype, which is impaired vision and difficult problems with their cornea. However, our mouse, we didn't see that phenotype in the heterozygous, which we expected to see.
This is due to the heterozygous expression expressing more of the mouse wild-type gene than the inserted human. To overcome this, mice models were made containing the homozygous gene for Keratin 12, and thus the phenotypic mutation was shown in the thickening of the corneal epithelium. Whilst in the human-type disease, it is thickening of the Bowman's layer that shows the phenotypic mutation, mice don't have this layer, so the disease is shown in the corneal epithelium.
So I think if we were going to make this model again, we would take maybe 15 nucleotides up and downstream from the mutation and change those to the human sequence, but that's the only change we would make. We wouldn't make all the other changes you see in figure 1. And I think if we did that, then we would get equal expression of the mouse and the human alleles, and we would see the disease phenotype in the heterozygous mouse.
Due to the inability to express the exact human disease type in the mice, it is not possible to produce a cure. This is because the therapeutic to produce a cure in the mouse-type model would not affect the human type in the same way. This is an example of a detriment of transgenics, as occasionally the disease in animals is too different to be beneficial to humans. Future studies will need to explore techniques to bridge the knowledge gap and make this a possibility.